Greetings everyone. In this video we are going to talk about mutated cell line analysis in Python. In this analysis we explore the genetic mutations present in WM115 cell line dataset. The dataset contains information on genes and their corresponding mutation types. WM115 is a melanoma cell line derived from human tissue and is commonly used in research related to melanoma, a type of skin cancer. We begin by examining the structure of the dataset, checking for missing data, and identifying genes with multiple occurrences of the same mutation type. We then proceed to visualize distributions of mutations, distinguishing between unique and non-unique occurrences. The goal is to gain insights into the prevalence and diversity of mutations within the cell line, which can provide valuable information for understanding its molecular characteristics and potential implications in cancer research. Today, we're delving into cancer research using data from DepMap Portal to obtain our data. But what is DepMap Portal? DepMap Portal is a vital resource for cancer research. It's a collaborative platform where scientists study the genetic dependencies of cancer cell lines. Let's start by downloading our file from DepMap Portal. Firstly, we open Google, then write DepMap Portal. DepMap Portal. And then we click on DepMap. Then on search, we write the name of our cell line, which is WM115. And then we go to mutations. And then we click on that arrow to download our cell line. And here we have it, our cell line. Firstly, we import our libraries. And then we read our data file. So step 1, data equal bt equal. So now let's check the column names. As we can see, we have many columns in our data. We have gene, chromosome, position, variant type, variant info, reference allele, alt allele, allele fraction, reference count, alt count, GT, phasing set, DNA change, protein change, and many columns. For the gene column, it is the name of the gene where the mutation occurs. And for the chromosome, is the chromosome number where the gene is located. And regarding position, is the position or location of the mutation on the chromosome. The variant type is the type of mutation such as single nucleotide variant, insertion, deletion, etc. And for variant info, it is additional information about the mutation, including specific nucleotide changes or effect on the gene. And for the reference allele, this tells us the original standard version of the gene at a particular spot in the DNA sequence. It's like the default setting. And for alt allele, this shows any changes or differences found at that same spot compared to the standard version. It indicates any mutations or alterations. Allele fraction, this gives us a ratio showing how common the altered version is compared to the standard one. Reference allele. Reference count, this counts how many times we see the standard version of the gene in our data. It's like counting how many times the standard setting appears. And for alt count, This counts how many times we see the altered version of the gene in our data. It's like counting how many times the changed set. And for GT, it is the genotype information indicating the combination of alleles present in the sample. This column typically represents genetic makeup of a particular sample or individual. It often includes information about the combination of alleles, variants of gene, present in the DNA at specific locus or location. And for phasing set, it gives information about the phasing of alleles, which is related to how certain alleles are inherited together. The DNA change, it tells us exactly what a change in the gene's DNA sequence. It describes the specific alterations, which nucleotides were swapped or added. As we can see, there are many different columns. So now, let's go for step 3. We read the first 5 rows of the data. So print data.head. Here we got gene, chromosome, position, variant type, variant info, reference allele, alt, allele fraction, reference count, alt count, and many other columns. As we can see, we have 63 columns. Now let's check for missing data. To get the missing data, missing underscore data bottom Now let's check if we have a missing data in our CellLine or no. As you can see, we have a lot of missing data. For the phasing set, it's 256. For the DNA change, it's 1. Protein change is 6. HGNC name is 3, and so on. And now let's go for step 5. Let's check for genes with more than one occurrence of the same mutation type. We are interested in gene and variant info. Paint with T-shirt top. Mutation 20 horizontal. Now let's print. And here we have it. We got the genes with more than one occurrences of the same mutation type. But what does it mean? For example, gene HLA-DQB1, it has 2 missense mutations. And for KPRP, it has also 2 missense mutations. And for TTN, it has 3 missense mutations. And for TYRO3, it has 2 mutations of frameshift variant and splice region variant. Now let's explain our code. For the mutation counts, equal to data.group by gene and variant info .size .reset index name equal to mutation count. This line groups the data by the gene and variant info columns and counts the number of occurrences for each group, and the size function counts the number of rows in each group. For the reset index name equal to mutation count, it resets the index of the resulting data frame and renames the count column to mutation count. The reset index makes the data frame easier to work with in subsequent steps. And then, genes with multiple mutations equal to mutation counts, and the mutation count more than one. This line filters the mutation counts data frame to only include rows with the mutation count which is greater than 1. It creates a new data frame, genes with multiple mutations, containing genes that have more than one occurrence of the same mutation type. And then the if condition, if not genes with multiple mutations, empty. This line checks if the data frame genes with multiple mutations is not empty, meaning there are genes with multiple occurrences of the same mutation type. In this case, print genes with more than one occurrence of the same mutation type. Overall, this code helps identify genes in the dataset that have more than one occurrence of the same mutation type. So, for example, if I have a gene that have missense mutation twice, in this case, this gene would be included in this analysis. So what does it mean? For example, this gene, which is PRAMEF1, here it has missense variant or missense mutation. And also here, the same gene, it has also missense mutation. So in this case, we have one gene which showed 2 missense mutation. So for example, in our analysis, if we get that the missense mutation affected 200 genes, this means that these genes are not unique. So it doesn't mean that we have only 200 genes, but maybe it means that we have 150 genes, but 50 of them are repeated, and are repeated with the same type of mutation, which is missense mutation or any other type. And now let's plot the non-unique genes based on mutation type. Firstly, we make group by mutation types and count the number of occurrences for each gene. And then we sort the mutation gene count series in descending order. And then we plot the non-unique genes affected by each mutation type. And now let's assign different colors to each bar. We make the colors based on the mutation gene counts. So if we have 10 types of mutations, we will have 10 different colors. And then let's plot the bar chart. color in all colors, and the label equal to genes. And now let's add the number on top of each bar for the non-unique genes. So I, V, 3, not the text, Y and C, also 1, TR, simple. The simple title, mutation type, and here we have it. And here we have it. We got a bar chart, contains of different types of mutations. Each mutation affected a number of genes, and it's written on the top of each bar. So for example, here for the missense mutation, it affected 240 genes, and for the stop gained, it affected 10 genes. And for the missense variant, and splice region variant, it affected 9 genes, and so on. But as we mentioned before, these genes are not unique, which means that here, for example, we have 240 genes, but some of these genes are repeated. So it's actually less than 240 genes. And maybe the same for the all other mutation types that affected some genes. So in order to fix this, we need to get the unique genes affected by each mutation type. And to fix this, we need to extract the unique genes. So now let's create a new column with the unique mutation types for each gene. And then we group by unique mutation types and count the number of unique genes. So and then let's group by unique mutation types and count the number of unique genes. And now let's sort the mutation gene count series in descending order. And then let's plot the unique genes affected by each mutation type. So plot dot figure fix size quarterly. And now let's assign different colors to each bar. Now let's plot the bar chart with the largest bar first. Now let's add a number on top of each bar for unique genes. We do the same for I, V, and here we have it. But this time we got the unique genes affected by each mutation type. Which means if we got a gene and the gene has two missense mutations, it will only appear one time. So as we notice, in the beginning, for the missense mutation, it showed that we have 240 genes. But because these genes are not unique, we did this analysis, and we got only 233 genes, which are unique and not repeated, and the same for other mutations. So here 233, 10, 9, and 8, and so on. So in summary, the analysis of WM115 CellLine dataset revealed several key findings. Firstly, we observed that the dataset contains various mutation types affecting different genes. Upon further investigation, we identified genes with multiple occurrences of the same mutation type, highlighting potential areas for further study. We then visualized the distribution of mutations, distinguishing between unique and non-unique occurrences. This allowed us to observe the prevalence of different mutation types and their impact on gene diversity. Write me in the comment section if you want to compare between two cell lines. Thanks for watching. I wish you enjoyed the video. If yes, then please don't forget to subscribe to our channel and hit the like button. Until next time, happy analyzing.